Hello. So here I am talking again. Once again, I'm doing a video and we're continuing on where we left off. We were talking about the electromagnetic radiation and the light. And we were also talking about the absorption spectrums and the action spectrums. And that's what we have a picture of here. This top picture is a picture of what we call the absorption spectrum. Okay. And we can see we have chlorophyll A, which is the darkest band here. And that's the main one. You can see that this is measuring the absorption versus the pigments down here, or the wavelengths. And I put VBGYOR to say that this is the violet range. Blue is around here. Green, I might have that skewed down a little bit. It should be maybe about right here. And then yellow. And then we have orange and red down here. And if you follow the darkest line, because remember we said chlorophyll A is the main pigment for photosynthesis. Well, chlorophyll A is absorbing the violet and in the violet and blue, mostly blue range and violet. Those are the high energy waves, right? But notice where it's reflecting. It's reflecting in the green and yellow. That's why leaves are green. And then also a little bit, well, not so much the orange down here because follow the dark one, but it is also absorbing red. Those are long wavelengths, which is very good, especially in the early morning and late at night when we have a lot of red colors that are being in our sky and stuff. Then follow the chlorophyll B. It's a little harder to follow these two lines, but chlorophyll B, notice where it's absorbing. It's absorbing in the blue-green like kind of area, slightly in the green, not as much. Absorbing in the blue and the green area, but reflecting in the green-yellow area, but it's also absorbing orange, okay? So we're kind of taking care of all the colors here. Then finally, we have the carotenoids. They're not really absorbing anything but the greenish yellow colors here. They're definitely reflecting yellow, bright yellow. And they're, follow the line a little bit here. They're absorbing a little bit of the orange, okay? That's a little confusing there. No, they're not absorbing any of the orange. My bad, you guys. It kind of like dies off right here. So they're not reflecting, or they're reflecting all the red, orange, and yellow. They're just absorbing like a little bit of green, a little bit of blue, and very little bit of violet. So you can see plants are mainly absorbing what I call the bit. Oh, that's what I did. That's what I did wrong. See, I forgot the I in there, the indigo. Okay, that's why everything's skewed down a little bit. How did I forget indigo? Okay, so you can see the BIV. Right? Blue, indigo, and violet are the main ones that are being absorbed. Green and yellow are the ones that are being reflected. And orange and red, a little bit of that is being absorbed. Okay, and that's the absorption spectrum. Now we look at the rate of photosynthesis. This is an action spectrum, and it's still versus the wavelengths. And what we notice is that photosynthesis happens a lot of times in the good old BIV area, the violet, indigo, and blue area, and also happens in the red and orange. But green and yellow light is not going to do that. Now, here's how this all kind of came about. This guy named Engelmann, he did this experiment where he put all this spirogyra, that's what this is supposed to be down there. It's a green algae that has chlorophyll that's all spiraled inside. They call it spirogyra. I'll write that here next to you. Such a cool little thing. Okay, and all this algae was in, and then they shone lights on it. They shone blue, indigo, violet, green, yellow, orange, red. They shone all these lights on it, and they noticed that, and they also it was in a fish tank-like thing. And in that fish tank, they had this aerobic bacteria. Well, the aerobic bacteria seemed to hang out in all the blue, indigo, and violet areas, or in the red and orange areas, but they didn't seem to hang out. That's because they're aerobic. They would want oxygen, and oxygen was only being produced in the red and orange area and in the blue, indigo, and violet area. So the bacteria didn't hang out in this area where they were shining the green and yellow light on. So this was a great experiment that kind of mirrored an action spectrum, which mirrored the absorption spectrum. Okay? So where is this going on in a plant? So we have this nice little picture here that's in your picture packet on photosynthesis. And here we have a leaf. And if we zoom up on that leaf, and you did plant tissues before, and when you did the cross section of a leaf, you had this middle layer, okay, called mesophyll. And you had these veins in there. We'll get to that in a second. And then you had the epidermis on the top and on the bottom. So let's get into detail about this. On the top is usually a waxy coating called a cuticle that protects the leaf from drying out. Just underneath that is the upper epidermis. Then in the mesophyll area, there's two areas. This area, which is packed closely near the top of the leaf, and that's why the leaf is more green at the top, is called the palisade layer. Palisade means like columns, and so that's what it made them think of, columns, okay? And then there's this spongy layer that has a lot of air gaps in it and stuff. This spongy mesophyll. So palisade mesophyll, spongy mesophyll, and then the vein. The vein is made up of your xylem and phloem, and we know how xylem and phloem transports water and sugars, okay? Then we have the lower epidermis, and of course on the lower epidermis, that's where we find the pores or the stomata. My favorite joke, what's a stomata? I don't know, what's stomata with you? The stomata are the pores, and on either side of the guard cells that open and close those pores and let the oxygen and carbon dioxide in. So this is where photosynthesis is going on at the tissue level, the organ level, the tissue level. So now we're going to zoom in on just a cell. This is like a spongy mesophyll cell. And we're going to look at these little green bubbles that are inside. Those little green bubbles are the chloroplasts. And if you notice the chloroplast here, and I'm going to flip to the next picture actually, because we're going to enlarge that chloroplast and take a look at it. But I want you to notice before I flip, it has an outer layer and an inner layer, and then these stacks. Okay, they look like coins, like all stacked up and like linked together. Okay, so let's zoom up on that and see what these parts all are. Okay, so here's the whole chloroplast, and we can see it has an outer membrane and an inner membrane, and there's like a space in between that is somewhat important, but the space that we're really more concerned with is going to be the thylakoid space. And these little discs are called thylakoids. And just like a cell membrane is a membrane around a cell, well, guess what the membrane around a thylakoid is called? The thylakoid membrane. And that thylakoid membrane is going to be very important in the light reaction of photosynthesis. Now, all the other space around outside the thylakoids is called the stroma. That's going to be very important for the dark reaction. I always call it dark reaction or the Calvin cycle, okay? So let's write that down on this diagram too. Thylakoid is where the light reaction will occur, okay? And the stroma is where the Calvin cycle will occur. Okay, and some other terms here, like the granum is just a stack of thylakoids. We got outer and inner membrane and the space in between. This is a real picture of a chloroplast, and they're showing you like the grana, and they're showing you the stroma area. So this is a real enlarged, like microscopic picture of chloroplast there. Okay, so that's the chloroplast. So now we're going to dive into the light reaction of photosynthesis. So what are we looking at here? So I need you to focus on one thylakoid. Like we're actually going to look at like one little part here on that diagram. Okay, so it can be the top part, it can be the bottom part, but one little thing. And that's what this is right here. Okay, so just try and get an idea that this little blub here, or bleb, or whatever you want to call it, that's like one thylakoid right there. Okay, and now we're going to look at the parts. Well, just like any membrane, membranes always have a double layer of lipids with proteins embedded in it. Well, so does the thylakoid membrane. Just like the cell membrane has a double layer of phospholipids with the proteins embedded in. So let's like even label those phospholipids. Phospholipids, right? And it still has that double layer. Remember, they're the barrier molecules. They're just like kind of holding the membrane together. It's really all about these proteins in there. And the proteins happen to be the pigments, okay? So they're the green stuff, all right? So we're going to focus on a few of these proteins in general, okay? We're specifically focusing on what we call the photosystems. Photosystem 2 and photosystem 1. It's ironic because we're going to start at photosystem 2 and then move to photosystem 1. So why didn't they call this 1 and then this 2? Because they actually discovered this one first. Some abbreviations for these: photosystem 2 is called P68. Photosystem 1 is called P700. Not really important, but another term that you can throw out there, okay? All right, and remember, stroma is all around the outside. So we're in one thylakoid, we're specifically in the membrane, and now we're specifically in this protein. So we're going to talk about this protein right here, photosystem 2. What's going on in photosystem 2 there? Well, there's a lot going on actually there. Think of it like an elevator. We're riding on the elevator, okay? But anyway, here's what happens. Light will strike that. Remember, light waves are coming in, right? And they're striking it. So photons, those waves are changing to particles, and those photons are transferring the energy. It's just like an ATP molecule transfers that energy into this little chamber. Well, water is also, you know how we water a plant? Water is also in there. It's diffused in and it's in there. And once it kind of gets in here, bam, that light starts splitting that water. And it splits it into three things: electrons, and we can see the electrons are feeding into this little like chamber here. Oxygen, which is just going to be a waste product. In fact, the next time you water a plant, take a deep breath, because as soon as you water a plant, and if it's in the sunlight, it's already splitting that water and releasing oxygen for you. And then also hydrogen ions. And we're going to see what these three things do. First, the electrons. They go on like a little journey of Leo says grr, oxidation and reduction. As these electrons go into this chamber, feed out from the water molecules and go into this chamber, they get excited. What does excited mean? Those electrons that would be at a lower energy level, remember the Bohr models, jump up to a higher energy level. So think of them as like, I'm excited, I got a lot of energy. Well, when they get that energy, they go skippy, skippy, skippy through all these other proteins. But while they do that skippy, skippy, skippy thing, which we call an electron transport, I can't spell here, tron transport, okay? While they go through this electron transport, they kind of lose their oomph, okay? And they're just like skipping. Think of it like, like skipping across a little shallow creek and you're stepping on all the stones. That's what the electrons are doing, but they're losing their energy. But while they lose their energy, they pump these hydrogen ions in. So now you got all these hydrogen ions that are building up in here, because not only did hydrogen ions come from water, but now they're being pumped out of the stroma and into here. So you got like a big buildup of hydrogen ions, while the electrons are just skippy, 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 skippy all along. But they lose their oomph and they get over here to this photosystem 1. Light strikes it again, boom, they go up, they get re-energized, okay? And we want energized electrons. Why? This is a light reaction. What is the whole purpose of the light reaction? I'll tell you right now. It's to produce this, NADPH plus an H plus, or NADPH, you can say it either way, and ATP. And you might be like, ATP, I thought that was like respiration. No, the light reaction's whole purpose is to make NADPH and ATP. And then, of course, oxygen is released as a waste product. So the whole purpose of light for the sun to shine on plants is really to make these two things. Now, you might be like, well, I thought the purpose of light was to make glucose and sugars. That's in the second half. Okay, so save up for the second half. So let's just go over this one more time, and we still mention this little protein down here. Okay, so we see water, it gets split into three things: electrons, oxygen, hydrogen ions. The hydrogen ions build up inside here. The oxygen just goes out. Okay, and the electrons go on a skippy, 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 skippy electron transport thing. They get excited, they get unexcited, they get re-excited. That's what the electrons do. So what about these hydrogen ions? Okay, we know what the electrons did, and we have these excited electrons, and we'll meet up with them again. But what about these hydrogen ions? Now, you see these dotted lines? Well, when the hydrogen ions, right, that are being pumped out here, and they're all building up in here, all these hydrogen ions, so much in there are going to go through something that will allow them to pass through. And that happens to be ATP synthase or synthetase. You can call it either one. And when those hydrogen ions go there, this thing kind of like churns around and makes ATP. Because remember, we want to make NADPH, which we really haven't seen that yet, and ATP. And we've now seen how the ATP is made in the light reaction. All these hydrogen ions that build up inside diffuse out, turn this little protein called ATP synthetase or ATP synthase, doesn't matter how you say it. ATP synthetase is what I'm used to. So ATP synthetase turns